today's conversation is with a world-renowned researcher from the University of South Dakota who specializes in the comparative functional morphology, biomechanics, and the physiology of movement. He has been a part of the chameleon hobby for decades. He's worked with more species than I can name, and he is an incredible resource to our community. I've never heard him talk about panther chameleons, ironically, and so this was a very unique opportunity to discuss panther chameleons with Professor Chris Anderson. This is pardalis. So hello, everybody. Today we have Dr. Chris Anderson here. He needs no introduction for anybody who's familiar with chameleons and has been in the hobby or has listened to Bill's Chameleon Academy podcast. Dr. Anderson has been a guest on there numerous times, but I personally am very excited to have him on to talk about pardalis and selective breeding strategies. It's a topic I've never heard Chris talk about, so I'm personally very excited for this conversation. So welcome, Dr. Anderson.
No problem. Thanks for having me.
Yeah, and so before we get into that main topic, one of the things I thought as I was preparing for this discussion, I heard a talk that you gave on chameleon anatomy, their tongues, the way that their tongues function, their skeletal structure, and it was awesome. So I'd like to give you an opportunity to just kind of give the elevator pitch and some of the things that you're working on for your research.
Yeah. So, anybody who's familiar with chameleons, of course, knows that they have this really unique feeding mechanism where they ballistically project their tongue from their mouth. And the mechanism by which they use or utilize to perform this tongue projection is highly specialized, and it's a very integrative mechanism that includes, bony elements, muscular elements, elastic tissues that act as springs. And effectively, the way that this mechanism works is that muscles in the tongue stretch and preload energy into these elastic tissues, kind of like drawing back a bow in a bow and arrow example. And then at some point during the feeding, that energy ends up being released from the system, and it powers the launch of the tongue. And what it does is it produces just this explosive performance where we have extremely rapid accelerations and therefore power outputs that launch the tongue up to 2.5 body lengths in smaller species to capture prey. And then muscle action draws the tongue back into the mouth with the prey item attached. And there's a whole number of features of this system that are super unique and specialized and intriguing, including the fact that this power amplification mechanism that they employ using those elastic recoil mechanisms produces the fastest accelerations and highest power outputs that have ever been recorded among any amniote. And so that's any mammal, bird, or reptile. It's with the highest accelerations and powers that have ever been released. And when I say power, what I'm talking about is not like a force measurement or anything like that. I'm talking about physics power, which is a rate of energy release. And effectively what happens is muscle is only able to release energy so quickly. It's about 1,000 watts per kilogram. And anything above that necessitates that there's this power amplification mechanism like these elastic recoil mechanisms. And chameleons experience powers that are about 14 times what muscle is capable of, what we ever recorded muscle being able to produce. And in addition to that, the prey prehension mechanism is also really specialized, where they have this kind of tripartite mechanism of holding on to prey, where they actually will mechanically interlock and grasp the prey item. They have wet adhesion that gives it kind of that sticky substance or that sticky feel to it. And then as they're drawing the prey back towards the mouth, they actually create negative pressure. So there's a suction component to it as well. And so the feeding apparatus and mechanism for chameleons is super unique and really interesting. And it's something that I study quite a bit in my lab, going back towards when I was getting my PhD work. And
Hmm.
so I have projects right now that are looking at specialization in chameleon feeding apparatus generally, looking at how some of these chameleons, when they're feeding on slow moving or uh, non-mobile prey items, will actually go up and rather than project their tongues, will actually just physically bite the prey item. So we're looking at
Hmm.
kind of like the evolution of these mechanisms from that standpoint. I have students who are, digitizing all kinds of feeding videos that we get from different chameleon species, looking at body size relationships and how smaller individuals and smaller species produce higher performance. I'm
Hmm.
doing a lot of work right now. I have a big NSF grant, National Science Foundation grant, looking at South African bradypodon species as well as some of the Southern African camellia species, and looking at thermal adaptation patterns in their feeding apparatus,
Yes.
because these chameleon species live in a really wide range of habitats, and living in these different habitats, they need to be able to perform at different thermal environments. Everything from Nemaquensis, which is living on desert sand dunes in Namibia with extremely high temperatures, to alpine species that are at high elevation, cold temperatures, and in these different environments, they have to specialize to that respective thermal environment. And so I have a lot of work going on looking at the South African bradypodon and that thermal specialization. So
Absolutely. That was one of the things that I really enjoyed about the talk was the fact that this mechanism
mm,
gives them an advantage over a reptile that relies on their muscles and relies on,
exactly.
and I
Yeah.
was like, wow, that's so cool.
Yeah, and
So
so the way that that works is, locomotion generally, which is powered by muscle contraction, fundamentally is you can look back and look at the muscle contractile physiology is all related to biochemical processes, and those biochemical processes, which are modulating that interaction between actin and myosin, the shortening of the muscles, creation of force, and everything, has a
mhm.
high thermal dependence, like any biochemical reaction would have. But when you preload energy into elastic tissues, the recoil of those elastic tissues, that's no longer a biochemical response, that's a structural response. And so that
Nice.
liberates that recoil from the temperature effects that we see in muscle contraction directly. And so effectively at low temperatures, chameleons are able to project their tongues out of their mouth at nearly the same performance that they are able to at warmer temperatures. Even though, and they do this by just more slowly stretching these elastic tissues. So they more slowly load that
Take
energy into those elastic tissues, but then that recoil is about the same, and the tongue launches off, at nearly the same performance.
Yeah, but I love that. I also love the other detail, which is it's not as fast at getting it back to their mouth,
Yeah,
right? So they
that's
can
very true.
they can
Yeah,
shoot it out, go get the prey, and then it's like slowly to get it back in. I
exactly. Yeah.
thought
So that actual prey contact happens really quickly, but
yeah,
then the muscle retraction that draws the tongue back into the mouth, that's still
still
affected by
does.
temperature, and
Yeah,
that's where that tripartite mechanism of prey prehension really comes important, into play, and is really important because at that point, if they can get a good hold on that prey item and hold on to it, they can draw it back a little bit more slowly and still be
still
successful.
exactly. That's really cool stuff, Professor
Yeah,
Anderson. Thank you. So, is there any way we could segue from this topic into
absolutely.
pardalis? Is there something cool about their tongues that we could
Just that
put?
they're chameleons and have that same mechanism. So
Yeah, and they're not quite as cool as the small ones. I was
not quite as
disappointed
high
to hear.
performance. Yeah.
Yeah,
I mean, absolutely, they're projecting their tongues out longer distances than these small guys, but
yeah,
relative to their
but
body
relative to their
size,
size, yeah,
it's a little bit shorter and lower accelerations, lower power outputs. But
yeah,
yeah, they're
yeah.
still impressive animals. So,
I know. It's like I still enjoy that. That's definitely a huge part of my experience with my animals.
oh yeah,
I love that watching them hunt. So,
for sure, for
um,
sure.
cool. So, I guess the one of the things that I've wrestled with as a breeder of the pardalis species is, um, I've come to it knowing some basic genetics,
Mhm.
Um, things like dominant traits,
Yep.
where if the parent has it and passes it to the child, you will see it.
Yep.
Um, you have things like incomplete dominant traits,
Yep.
where you get variance. Like if they only get it from one side versus getting it from both sides, you're
Yep.
going to see a difference between a single gene and a double gene.
And the
Uh,
translucent genetics for the veiled chameleons, that's basically a codominant trait where you have the kind
oh,
of expressions as being for both parents, low expression
and low
being dominant,
expressions.
and then
Wow.
full recessive is the standard not expression.
So I was always under the impression that translucence was a recessive trait, but it's codominant.
Well,
It's
that's why you see the low expressions versus the high expressions. Is it is
uh,
it's actually a codominant trait. I mean, that's what the numbers,
that's
at least
cool.
the early breeding and so forth, where we first got the genetics in, was suggesting is that it's codominant. But again, nobody's really ever done any careful
Statistical work on it.
quantitative
Yeah.
statistical work on it to really show that. But you know the fact that you see some of these individuals that just have it on their feet, on the tip of their toes and so forth, and then when you
Hmm.
breed those together you get some of those, but then also some
You
high
get like a
expression animals, is suggestive that there's some additive effect which would indicate that codominance is likely at play.
yeah, and you don't see the kind of thing where a translucent produces zero
Yep.
translucence, which is
Yep.
what you would expect from a recessive.
Yeah, exactly.
So okay,
Anyway,
cool.
sorry, yeah,
That
not to
already
segue
we no, we
horribly
already
into
started
pardalis.
with something I didn't know. I'm glad that
There you go.
because I've said that it's a recessive trait before, I think
Mhm.
publicly, and so now I'm gonna have to go back on that.
No
That's that's
worries.
that's cool. I've never bred veiled, so but yeah, so that's a good introduction to I guess what I would like I've referred to as Mendelian traits. Some people are
Yep,
not familiar with that terminology, but the way I see it is those are simple relationships. It's kind of a one to one where
definitely.
if the gene is present, you see it, or you
Yep.
don't, and you need two to get the outcome you're looking for.
Yep. And it
Um,
produces some pretty basic percentages in the offspring. If you statistically look, if you're dealing with one gene, oftentimes it's going to be 25%, 75%, or 25%, 50%, 25% if it's a codominant and so forth.
exactly. So
Yep.
your ratios that you should be seeing should be pretty clear cut.
Exactly.
Even if you don't hit on one clutch over a long period of time, you'll start to see that.
Exactly.
Uh,
Yeah. Over time, if you take it all together, they basically work out to those type of relationships based on that, simple Mendelian Punnett square, if you will.
Yeah, exactly. And so one of the things that I thought was cool about pardalis or breeding pardalis for a specific color is it's not that simple.
Yeah.
Um, and not
And
only
that
that,
simultaneously makes it interesting and exciting, but also really frustrating.
yes, yes, for customers, I apologize for customers who have been
Yeah.
disappointed because we've all been there.
Yeah.
But it's like it's very challenging because we see more of a like a continuous
Mhm.
spectrum of results that average around
Yep.
the sire and the dom's lineage.
Yep.
Um, can you talk to us a little bit about that? What are some of the ways that that can happen in
Yeah.
nature?
So again, the genetics of panther color really hasn't been studied extensively. We don't know a whole lot about what genes are controlling it, how many genes they are, how many different alleles there are at any given gene. But what is fairly clear is that it's being regulated by multiple genes. It's a polymorphic type of trait where we have many different possible alleles, likely multiple different, parts of the genetic sequence that are affecting the color that's exhibited by the males and females. And what that means is that you start getting an exponential number of possible combinations that can be expressed. as you start doing crosses and out crosses and breeding. And so the goal, of course, if you're a breeder and you're trying to produce certain types of genetics, is to reduce that variation, which basically would be moving towards specimens that have like genetics and similar coding going on at every single one of these genes in order to produce a typical goal for an offspring. But
Hmm.
getting there is incredibly complicated because there's so many combinations, so many individual genes that are regulating it, and it can be extremely challenging and throw a lot of different permutations into the equation.
Yeah. And my favorite one is sexual dimorphism.
Yeah,
So,
yeah.
so you're 50% blind every time.
That's exactly right, and so that makes it even more challenging because when you're dealing with a female, you can look at the rest of the clutch that female came from and look at the males and get a little bit of an idea of the genetics that's within that clutch. But if you only have, say, a single male from that clutch to look at, you don't know what happened to the rest of the males, don't know what they look like. You
Hmm.
still are flying blind to a certain degree because
Yep.
you don't know what that female has genetics wise compared to that other individual, because that clutch could have itself been extremely diverse in the phenotype that it expressed, and so you may have a female that has next to none of the same genes or combinations of genes as one of those other males in that. And so that makes it incredibly challenging. The
Yeah.
other challenge there is, particularly with wild caught females, a lot of the times we have multiple paternity where, a female can breed with multiple males. And then you also have that combination of, not only do you have, you know, the two parents, you have multiple males that could have
Yeah.
given or been the father for that individual, and that could have been different than the other male in that clutch. You also
Hmm.
have sperm storage, and so you don't know if it's the male that you observed breeding, or if you had a previous breeding with another male and that sperm is being used to fertilize the eggs. And so there's a ton of permutations, and when the females don't express these colors that the males have, you only have part of the equation to really even start factoring in what the possible combinations might look like.
Exactly. And in captivity, that's even a challenge, given that
Absolutely.
people will be pairing multiple sires to the same female
Yep.
without waiting long enough for her to actually be laying infertile or
Absolutely.
other things. So I
Absolutely.
that's one thing as a breeder I track is like, hey, has this female been bred
Mhm.
before?
Yep.
Uh, what did that guy look like? Like.
Exactly. And so then you get situations where, particularly if it's a female that maybe was unproven or something, that you don't know yet the first time you breed her, what the clutch mates looked like to confirm if it was a certain locality. If, for instance, it shows that it wasn't the locality that you thought it was, and then subsequent breedings you go back and breed her to the same locality that her clutch mates showed that she was from, you still can get into a situation where locale hybrids or
Mhm.
locale crosses because of that sperm storage, and it can
Exactly.
be a very challenging proposition to try and track and maintain known genetics, and particularly shooting for a specific color combination goal.
Yeah, and you called it a phenotype earlier. Can you elucidate what that means
Yeah.
for people?
Yeah. So when I
Yeah.
talk about a phenotype, basically what I'm talking about is the ultimate expression of all of the underlying genetics and environmental factors that go into producing what we're seeing expressed in that animal. And so that's the color combinations, the size, the shape of that animal, any basic trait that we can quantify visually, and or quantify, measure in other ways, that would contribute to the phenotype as opposed to the
Phenotype, yeah.
genotype, which is going to be the underlying genetic, coding
Driver.
and
Yeah.
combinations that individual has.
Yeah. Thank you. I've used the term, and then I realized that some people are like, phenotype, what's that? Is that a
Yeah.
morph? And I'm like, no, it's not a morph.
Yep.
Uh,
It's basically whatever that individual is showing, whatever that
yeah,
animal expresses as its color, its pattern, its body size, its head shape. It's any of those things would be the phenotype as opposed to the underlying genes.
yeah. And those traits are things that we often associate with locale in pardalis, right? Like we'll
Yeah.
have a collection of phenotypes that we've observed in that location, and
Yep.
we use that to say, oh, this is in the hobby
Typical.
what we see, yeah, what we
Yep.
see as typical in this location.
Exactly. That's exactly
Yeah.
right.
And um, one of the things that I wanted to cover a little bit too is there are differences across locales. And so sometimes when people hear about like polymorphic traits or polygenic traits, um, how how does that work in the context of the locale?
Yeah.
Um, yeah, because
Exactly.
sometimes
So
people will say, "Oh, it could be anything," right? Like you because it's polygenic, right? So
yeah,
you can create literally anything, right?
exactly. Yeah. So anytime we're talking about something that is a phenotype that's closely associated with a specific area, geographic area that an organism comes from,
Hmm.
we're looking at a situation where the gene frequencies at that location are higher in certain areas, or certain genes have higher frequencies in that geographic
Then
location
that
than they do in other locations. Code a specific gene or be contributing to a specific gene that are in low concentrations at that specific location. And so if I just really simplify it, oversimplify it, for instance, at one location, at one locale, there could be a high proportion of individuals that are blue in coloration. Whereas, and we could say that the gene frequencies for whatever genes that are coding for that blue coloration are at higher concentrations, a higher levels in that population than in a population that doesn't show very much blue and say is very pink. And
yep.
in that location, the gene frequencies are very different. But because we're talking about a polymorphic trait that has multiple different gene locations in their genome that are contributing to the ultimate color, there is some variation. I mean, you look at two individuals that can be caught in the exact same tree, and they will show some variation. They're not going to be perfectly identical. And that's the same thing that we see in humans. Just because you're from Minnesota or from Texas, you're not going to look exactly the same. And that's because we have inherent variation in our genes that allow for us to have individual characteristics and traits.
Hmm.
But effectively, when breeding occurs within a location, a specific location, when you're you end up with kind of a bell curve of possible permutations where you have kind of the normal average that you would expect to see kind of most frequently at that location. But then as you deviate from that, you can still see some variation, but it becomes less and less frequent. And
Hmm.
effectively, what that does is it makes it so that, you have a typical expression of the color and pattern genetics at a specific location, but you still have some variation that allows for some movement, in one way
Within,
or another within that population.
yep.
Now, it becomes a little bit more complicated when you talk about pardalis because many of those pardalis are not geographically isolated from each other. And so, it's really nice and clean when it's just like Nosipe, you're talking about an island, and then there's, an ocean that's separating it from the other island and so forth. And that really interrupts gene transfer between those populations. It really reduces them.
Hmm.
But what you see on the mainland of Madagascar is you have, Ankarame, you have Mbilabe, you have Mbanja, you have Diego Suarez, and then coming down the other coast, you have multiple localities that are geographically spaced apart from each other, but there's
Hmm.
pardalis between them as well. And so the question is well, how do you maintain this standard, this norm at one specific locality when you have the potential for gene flow between adjacent populations? And really, what that gets starts getting down to is, are there any types of barriers between them that start to slow any of that gene flow? And it may
Hmm.
not stop the gene flow. And even on those islands, you don't stop the gene flow between adjacent islands, because you can have rafting events. You can have an animal that ends up swimming somehow and makes it inserts its genes into the population. You still have some natural gene flow. But if there's anything that slows that gene flow, and that can be, a river, that can be, a
Mangrove
major
forest.
highway, it can be, a change in elevation where you have a rapid, decrease or increase in elevation as you go from one place to the other. That
Hmm.
is enough to start to slow the exchange of genes between these populations, where you start to get more distinct expression of those possible color genetics at these two different locations. And so the complicating thing there is, you say, okay, well, if it's not stopped, if it's just slowed, you would think, oh, you could just have any combination of colors. You could have any genes. You could have an Encarame that pops up, one of these pink panthers that pops up in Mbanja.
Hmm.
And the important thing to remember there is, we're talking about relatively small animals. We're not talking about an animal that's going to up and move 50 kilometers and then breed, in Bilibay with the animals up there. We're
Yep.
talking about animals that are moving, dozens to maybe 100 yards over the course of a season, because they may be moving around, but they're moving around in kind of a circle around their general area.
Yeah.
um, and that means that for their genes to eventually reach Mbanja, say, from Nkarame, that individual is going to have to move a little ways towards Mbanja. It's going to have to breed with an animal there. Those offspring then have to start, maybe one or two of them move a little bit further, and
Hmm.
in next generation a little further and a little further. And what you do is, as that happens, you start to water down those firm and Karame genes as you get closer and closer to Mbanja, because you're encountering animals that are a little bit further and don't have quite the same gene frequencies that they have in Mbanja. And so
Yep.
over, hundreds of thousands or millions of years, you might have that gene expression where you have a couple genes from Nkarame that end up in Mbilivae, but ultimately that's going to have a fairly negligible impact in many cases on the gene frequencies of that population. And so, we have continuous populations, but the gene frequencies and the exchange of genes is quite slow. And that's what helps to maintain the specific phenotypes that are expressed at different locations.
Very cool. And I read the 2015 paper by Gerbic et al. I
Yeah.
think that's probably one of the more popular
Yep.
research papers on the species.
Mm.
Um, how does that play into this discussion?
Yeah. So, what happens over time when you have distinct populations that should start to show different gene frequencies than other populations, you're starting to get population structure at that point, where you're starting to
Yeah.
see distinct differences between populations in some of their genetics and so forth. And I mean, this makes sense. There's certain parts of the world that people tend to have, blonde hair on average. Certain parts of the world where you see more, dark hair and so forth. It is a similar type of thing. There's population structure there where you have some distinct characteristics that are associated with where those people are from. In this case, these chameleons are from.
Hmm.
The question becomes, when do those differences in these chameleons become significant enough to warrant considering them to be different subspecies or different species? And
Yeah.
that differentiation between populations is separation, in those species that's maybe on that continuum towards eventually maybe separating into distinct species. But it's a gray area where that line actually occurs.
Yeah.
And so you can have population structure, but still have these populations be from the same species or same subspecies, for example. Now, this study that you referenced basically looked at some of the physical characteristics as well as the color repertoire of pardalis at different locations across Madagascar, across their range in Madagascar. And they found
Hmm.
distinct population structure where certain populations exhibited certain traits at higher frequencies than other populations, and effectively were able to kind of divide the species into a certain number of geographic localities, if you will, that are quite distinct from each other. And that makes sense, because if you look at the pardalis and the different localities that come in, you've got some pardalis localities or regions with multiple localities that express a lot of red or
Hmm.
orange, and then you've got
Yep.
some that express a lot of blue as their base colors, and others that express a lot of yellow, and others that have this pink coloration. And so it makes sense if you look at a pardalis. Oftentimes, if it's an import, you might not be able to say, oh, that's a Simbava versus, oh, that's a Marwa and Cetra, but you can very
It's East
oftentimes
Coast.
put it into a general region based on some of those differences.
Yep.
And that's effectively what their analysis showed. However, in their discussion, they very briefly noted that these could be eventually warrant separation of species. And then subsequently, when that article was covered in the popular media and press, and they did interviews, they really harped on the fact that pardalis now are multiple species. This is evidence of
Hmm.
multiple species, when in fact they
Yeah.
didn't show that. They didn't have evidence to support that. They didn't really even push that in their paper, that that's what their findings were. And so it got blown out of proportion that pardalis are multiple species, when that's not what they showed. It may
Hmm.
be that they are moving along the process towards ultimately at some point getting to the point where you have distinct species across the current range of pardalis, but they're all pardalis at this point, pending significant additional data that supports separating them.
Thank you. Yeah, I remember when those news reports came out, and it was
Yep.
like, "Oh my gosh, are we actually creating hybrids
Yeah.
between subspecies when we do cross locale pairings as a breeder?"
Yep.
Um,
Yeah.
and that for the hobby had clear implications for, responsible and ethical management.
For
Um,
sure. And,
yeah.
I would say that we still have some of those same considerations, even when we're talking about, localities, and maintaining kind of the wild forms of what we see, for the animals across their range. I don't think it rises to the same level necessarily as when we're crossing species or something like that. But
Hmm.
it really then comes down to your personal goals of what you're trying to achieve and what you're trying to look at. If you're trying to, argue that your efforts in breeding them in captivity have any type of conservation relevance, and you're hybridizing localities, you're shooting your argument in the foot, because you're not
Yeah.
actually maintaining the species as it is reflected in the wild. So
Hmm.
there's an issue there, and theoretically the same could be made. Same argument could be made if you're really trying to selective breed within even a single locality for specific combinations of traits. That's not really a breeding strategy that is focused on maintaining the genetic diversity of a specific location. That doesn't mean that it's bad.
Hmm.
People can do that, and that's fine. But it ultimately just is practices that kind of are, you need to decide what it is you're looking to do and to achieve. And if you're looking to work with ambilobe or ambanja and really produce a specific set of traits that are seen within that species, and really see what that genetics is capable of producing, that's great. you
Yep.
know
Yep.
it's just a matter of knowing what you're trying to do with your animals and your breeding efforts.
That yes, and I personally have come at it from that perspective of working within the ambilobe locale,
Mm,
trying to produce yellow body
mm,
animals and then trying to produce red body animals and treating those two populations as separate projects, not crossing them, even though
yep.
I know that they're likely to breed in the wild. Um, in captivity, I have the breeding goal of consistently producing yellow over here and consistently producing red over there,
Yeah.
knowing that those are both wild phenotypes.
Yeah, yeah, exactly. And so basically, what you're working on is producing multiple pure lines, relatively pure. I mean, they can't be perfectly pure. Multiple pure lines that are representative of the diversity that's within a specific locality, and that's fantastic. That's great, effort to make.
Yeah, and I'd say one of the big challenges that I have is how to communicate that to a customer,
Yep.
um, because even though I have that goal, I'd fail.
Yeah.
Um, it's so I don't want people to be disappointed.
Mm.
I don't want to overpromise, but
Yeah.
at the same time, I want to provide enough information for them to make an educated decision.
Yep.
Um, what are some of the things that you would look for as a sophisticated customer of Pardalis?
Yeah.
Um,
So one of the things that if I was looking for a specific locality or a specific expression
Phenotype,
of
yeah.
the phenotype from a locality, I would definitely want to look at bloodline history, and so some information about what the sire, the father of this clutch, as well as the mother and her clutch mates looked like, and trying to see kind of how diverse maybe the clutch in the female was in the genes that were expressed or the phenotypes that were expressed. How similar maybe the male siblings of that female are to the sire and dam siblings. For the female, the
On the
mother of that clutch as well,
Mhm.
and try to kind of see how much variation you're seeing in what's being expressed as you go back a couple generations. Um, Ultimately, the more variation you see as you go back into successive generations or looking into a single clutch of one of the parents or grandparents or something like that, that's suggestive of more possible permutations for the phenotype to express. And so the more consistent what you're seeing with those previous genes or those previous generations are, the more likely subsequent generations will show similar type of genetics. And so, yeah, I mean, if I was looking for something really specific, I'd want to see multiple generations of, or as many generations as possible of, sire and sibling and
Siblings.
yep, and so forth, to start to get a picture of what diversity I might expect in that type of pairing from those animals.
And why did you pick the female?
Because the female, is that loose cannon. You don't really know what that female it's herself is going to show, and so you have to look at other individuals in the clutch, males from that clutch, the sire and the dam of that female, and the clutch mates of the dam of that female, and so forth, because the male is showing a lot more information about the underlying genetics that he has to pass on than the female is having. The
Hmm.
male, you can look at that male and kind of get a pretty good idea of some of what he's going to contribute. The
Yeah.
female, you really can't unless you look at some of the siblings, the parents, and the parents'
And
siblings to start piecing that together.
as she ages, her children, right?
Yep, exactly. Yep, that's
Yep,
exactly right. As she's had more clutches over the years, what she actually produced previously as well.
exactly. And I've used the term to try to simplify it for people. On the male side, what you see is what you get.
A little bit, yeah,
And it's not that simple. I understand that, but
yeah,
it's like you look at them. That's
mm,
the most important information is
yep,
what you can see on that animal. Because I often will have people looking up his lineage, like two generations, and being like, "I don't want that." And I'm like, "No, that's stop,
yeah,
like stop. You look at him, please."
yep,
And that so I've tried to simplify it. I know that it's not that simple,
Yep.
but from an information gain perspective,
Yep.
like you gain a lot more information from what you can see on him versus say
Exactly.
like his grandfather on his mom's side.
That's exactly
Um,
right. Yeah, I mean, you have a much better view of the genetics that are going into encoding color by looking at the sire than you do the female. And
yeah. Yep.
the parents of that sire may have had the capacity to produce some variation, or may have themselves had slightly different colorations, but that's not as likely to be passed on from a sire that's not showing those colorations as it might be from a female who you have no idea what her male colorations would have been.
Exactly.
Yeah.
Yeah. And and on the female side, one of the things I'm a data scientist
Mm-hmm.
as my day job. So I think of things in like prior distributions to
Mm-hmm.
estimate what you're going to get. So you're collecting as much information as you can to
Bayesian
to
stats. Huh?
yeah, yeah. So right, it's like you're trying to put together an expectation for
Yep.
what you think she could have, right? And so you're
That's right.
just gathering as much information to get an accurate estimate and knowing that it is a distribution and she could be on
Yep.
the tail and all
That's
that, right? But that's you're just that first part is just get that estimate, and then after that s- update that information with the males you prove out from her,
Yep,
right?
exactly. So that improves your priors so that you can start getting a better picture of what subsequent breedings might look like. And that's why you mentioned if it's a female that's produced multiple clutches, looking at the clutches that she's produced will give you a lot more information for you to make an assessment of what her offspring will likely look like moving forward.
Exactly,
Yeah.
because you and I try to explain this sometimes with breeders who are getting started. I'm like, you use that lineage information just to get to select your female,
Yep.
right? But once you have her and you decide you're trying to figure out if you want to keep that genetics in your project,
Yep.
try to forget it. Like try to
Yeah.
forget that. Focus on what you're proving out from her because that's the reality. Like you're guessing at the beginning when you're selecting her, but then
Mhm.
from that point forward, it's all about her males and
Yep.
her male offspring and what she actually did
What she's
produce for you.
actually doing. Yep. Yep.
Exactly.
Exactly.
Yeah. And that's tough for people. I feel like they still stick to those lineage pictures and can't get off that prior.
Yeah. Yeah. I mean, and some of those previous lineage pictures can be informative to as well. Particularly if you're worried that somewhere back in the background of a certain individual, there might have been genetics
Mixed
from another
locale.
locality that were introduced. Going back and looking at that at before back crossing into whatever locality that's being advertised has occurred, you might be able to get a little bit more of a picture of that if you're familiar. But yeah, once if you're looking for specific traits, specific characteristics within a locality, looking at the offspring that female has produced is really going to be a lot more informative at that point.
Awesome.
Yeah.
Yeah. And um one of the other things that I've been trying to wrestle with, besides the female challenge, which I think is the most fun
Mm.
thing about this species, um, is things like genetic distance.
Mm.
So on the one hand, we have a goal, like
Yep.
we say in my case, it's producing yellow.
Mm.
Um, you're trying to manage the uncertainty on your female side, but you also want to maintain genetic diversity. Um, and so what what are some of the things that um, could possibly help inform where that distance could become problematic, or what's a reasonable
So
distance
you're
to work
so you're
with?
asking about relationship,
Like,
how closely related individuals are breeding back. Yeah.
yeah,
So this starts getting into questions about inbreeding versus line breeding, and
hmm
when when is it deleterious to the offspring and to the animals versus when is it enhance the goal without having the serious risk of deleterious effects. And so ultimately, what you're concerned with when you're thinking about these type of questions is whether you're going to increase the likelihood of harmful genes being passed on to subsequent generations, because most individuals in any population have certain genes that maybe aren't beneficial or could even be harmful if they're expressed in their homozygote, their condition, that they've got received that same gene from the male or the female, or potentially it could be multiple genes that are interacting where if you get a certain combination, you start to have some type of deleterious effect.
Yep.
And the chances, the likelihood of those combinations, those non-beneficial combinations appearing in an offspring increases when you are breeding individuals that are really closely related back together, because those individuals are going to be more likely on average to have those deleterious genes that they could pass on than a random sampling of the population.
Yep.
So the kind of ideal type situation is random outcrossing within a population that would reduce the chances of those genes, those potentially deleterious genes, coming together in an offspring. But when you do that, you're also fighting yourself with trying to select for certain colors and so forth.
Yep.
And so if you're trying to select for a certain trait, you necessarily are not performing random pairings at that point. So you want to select for individuals that have similar or have traits that you are shooting for and breed those together. But the chances of similar looking animals being more closely related also starts to increase because of just genetic frequencies and so forth. And so it's a little bit of a balancing act. Now, in many cases, if you really wanted to select for something quickly and just get the that specific gene, reliably in offspring that you could then breed, you might breed siblings together. And this has happened in many cases when there's a new morph or something like that that's appeared, or potentially breeding an offspring back to a parent that has that.
Yep.
And that's where you really are starting to run into the biggest risk for those deleterious genes presenting themselves in the homozygote condition in the offspring.
Yep.
You then have differing levels of back crosses that or out crosses that you can perform, where you maybe have an offspring that is expressing traits that you really like, and then you could breed it to an unrelated individual that maybe has some of those traits but doesn't have a known connection genes wise otherwise back into that individual to increase the diversity of some of those genes, and therefore hopefully water down some of those potentially deleterious effects. But the further that relationship is from the individuals you're trying to select those traits for, the more you're also fighting your own efforts in getting down to a specific expression. And so oftentimes what people will do is they'll breed siblings or potentially they'll take cousins or something like that, where they have say they select two males from the lineage that is of interest. They'll breed both of those males to females from another lineage that's pretty good that they like, and then the offspring of those two pairings they might breed back together. So there's certain things that you can do to introduce new genetics, but still try and hone in that focus on specific traits that you're looking at while trying to avoid some of those deleterious effects. And it really just comes down to, at that point, the breeder needing to be careful not to select individuals that are clearly weak and exhibiting potentially deleterious type traits, and breeding those back into that population, particularly in close subsequent close subsequent generations where you
Yep.
might see some of those. When you do start seeing that, that's when you start seeing certain traits being weaker than others because there's just been too much of that inbreeding that's occurring.
Hmm.
Yeah, so line breeding versus inbreeding is basically inbreeding is typically sibling to sibling, sibling to parent type interactions. Line breeding is when you are doing outcrosses and then taking offspring from some of those outcrosses and breeding them back, and introducing new genetics to maintain some of the strength, but still breeding closely with that lineage within that lineage to try and maintain those
Maintain it.
characters.
Yep.
And if it's done carefully, line breeding can be done fairly safely, but there is that interaction with the ethics of the breeder as well, and making sure that the breeder is ethically choosing which individuals to breed, not necessarily exclusively looking at whatever their trait is and not paying attention to other symptoms of yeah
Yeah, like eye,
problematics.
eye and tongue functions, shape and size.
Yep, yep,
Yeah,
exactly. Neurological issues, yeah, growth, yeah, things like that.
yeah, yeah,
So,
absolutely. Um, that's great. I mean, the rule of thumb that I often think of is these negative outcomes are often, I think of them as a recessive outcome, right?
mhm,
And to estimate the likelihood
yep,
of something bad happening. Um, and the way that that works is you've got to get it from both sides.
yep,
And if it's siblings, then there's a 50% chance that each of them have it, and
yep,
then a 25% chance that their offspring would would end up having the deleterious impact. And so it's always like a nonlinear decrease
yep,
as you add a generation of distance.
yep,
And the estimate that I've used, and I know it's not a discrete thing. It's kind of when you simulate it, you can get different ranges of outcomes, but
mm,
somewhere around 6 to 7 generations is when you drop below a 1% chance of a deleterious impact if it is a
mm,
recessive
yep,
trait. Um,
yeah,
so that's kind of the rule of thumb that I'm like, okay, well that's low enough.
yeah,
Um,
yeah.
below 1%, because now we're getting into like normal kind of things that can happen in
Random
a
chance.
clutch. Yeah, like
Yep,
random chance versus it being actually due to the shared lineage.
exactly.
Um,
Another thing to keep in mind too is that it seems like certain organisms have maybe a higher propensity to have these deleterious traits kind of show up in response to inbreeding than others. In mammals, for instance, it seems to be a big issue, but in reptiles, it maybe seems to be a little bit less problematic. And you can kind of see that a little bit by looking at the size of the founder stock for many species or populations. And so if you look at like the introduced Jackson's chameleons in Hawaii, if
mhm.
you go back, there's some records that suggest that the original founder populations was based on like fewer than a dozen individuals. And if that's the case, you have a population, a really large and flourishing population in the Hawaiian Islands that is not particularly diverse in its founding genetics, but
Mhm.
is doing well by and large. And there's some differences, of course, between the Hawaiian populations versus the Kenyan populations that, and maybe some issues with how robust they are or something, but by and large, they've done pretty well. The similar case is the veiled chameleon in captivity. Turns out the veiled chameleons in the US and in Europe are not from a hugely diverse founder stock that was originally imported from Yemen and Saudi Arabia.
Mhm.
There wasn't that many founder individuals that have produced the population that we see in the wild, or I'm sorry, in captivity, including the Florida populations. And those Florida populations that are introduced, it's not like we've started over with freshly wild genetics. Those are
Nope.
have some founder effects from whatever individuals from captivity that were already a limited gene pool that were establishing
Mhm.
those populations. And so, you know,
Yeah.
um, with chameleons in particular, there's a number of instances that we can cite that show that limited founder populations, and inherently what will end up being some inbreeding, maybe doesn't have as many of the negative side effects as we expect to see in like mammals or other instances.
Very cool. And
Yeah.
and one thing that's also interesting about those small founding groups is that it's also less likely that a negative, like a bad gene is in there, right?
Yeah.
The
Yeah. Otherwise,
so
it
it's
wouldn't
kind of
be
like a
successful. If you have two individuals that have deleterious genes, and those are going to be your founding population, there's going to be a high
it's not
prevalence of those deleterious genes, and that population ultimately is not going to do well. But if you have a
hmm
population that's done well, that it's suggestive of the fact that, okay, there's probably not a very high proportion of those deleterious genes.
yeah. And in my field, we call that survivorship bias.
Uh-huh. Yep.
That's like you see the population that succeeded, but you don't see all the ones that failed.
That's right. Yeah. Exactly.
So it's
Exactly.
like how that's a really interesting question to get into, is like how, as you step back from the one example of a success, like how many of them failed, and was
Yep.
that due to the fact that the deleterious gene was actually present,
Yep. For
right?
sure.
And yeah, so that's a really cool topic.
Yeah.
um
Yeah. Definitely.
and and for me as a breeder, I've always I've looked at it and said, okay, precautionary principle
Yep.
prevails here, and
Absolutely.
just do your best to do unrelated stuff while trying to achieve your goals,
Absolutely.
and outcross to keep the genetics
Yep.
diverse, and just eat it when the customers are unhappy, because they're like, hey, this one wasn't red, or this one wasn't yellow, and I'm like, okay,
Yep.
well, I have to have genetic diversity. I can't just
Yep.
inbreed everything to get the exact result that we want.
Yeah, absolutely. I mean, best practices are definitely, for the most part, you need to be cautious and not just rely on. Well, it worked well with veiled chameleons in captivity, so it's not always,
So
like you said, it's not always going to necessarily end up like that.
yeah, yeah. That's awesome. The other thing, I think, just thinking about the female challenge that
Mm.
I find difficult personally, is the way that we think about things is in discrete outcomes.
Yep.
Like we're looking at one picture of the sire. We're saying
Yep.
this is the sire, and then you look at one picture of the dom sire, and you have those two things kind of anchoring you in your
Yep.
head.
Yep.
But we really need to think about that female on a continuous spectrum,
Yep.
right? Not discrete. It's not like 75% these two things,
Yep.
right? It's she's somewhere on that spectrum.
Yep.
And if she's on the tail and you mix that in, then
You've increased
you're then
variation. Yep.
increased variation, and then the outcome itself, you could actually have a yellow sire, a yellow dom sire, and then that female hits on the red side of the spectrum.
Yep.
And when you have red and yellow, now your average is going to be somewhere around orange, right?
Yep.
You're
Yep.
not even going to your average outcome is going to
Yep.
be an orange looking chameleon, some being red, some being yellow,
Absolutely.
and people are like, yeah, but it came from two yellows. How is this possible, Chris?
I mean,
How's this
so
possible, Mr.
so
Anderson?
this is what happens when you have polymorphic traits, where you have, you can have multiple individual gene locations that are contributing to the yellow coloration, or to the
Mm-hmm.
red coloration, or whatever color it is you're looking for. And so you can end up with a situation where, say, the dam sire expressed yellow, but had some variation in those where it wasn't necessarily a homozygote expression or homozygote condition, where all of those genes for yellow were both copies of the gene were the yellowest yellow. There was some
Mm-hmm.
variation there. It
Yeah.
still ultimately came together to produce yellow. But then you also have the dam's dam, and if there's any variation there, and when those come together, you have a situation where all of a sudden you've mixed up the genetics from two different individuals. You can get different combinations from, say there's four or five individual genes that contribute to yellow. And I'm
Hmm.
guessing, I'm not even guessing, I'm
Maybe
just using that as an example.
exactly.
Um,
Yeah.
but you could have,
Theoretical.
you know, all four or five of those genes. You could have the dominant gene that's expressed in that male passed on. but you
Hmm.
could have some of them that have, one or two of them have the dominant gene, and two or three of them have the recessive gene. And then all of a sudden, if you have an individual that has a different combination of dominant and recessive genes across those four or five genes, that's going to produce a different color. If that it's a male and that color is being expressed than it would have previously. And similarly, you could have two yellow individuals. Let's just say that for a second that the females expressed the color that the males do. You could
Hmm.
still hybridize two yellow individuals but have very variable expression in the offspring, because they could be expressing yellow because of the first two of those yellow traits, whereas the other
Hmm.
individual could be expressing yellow because of the last two of those yellow traits. And then all of a sudden you mix that up, and now you don't have any of those dominant traits that are being expressed in that offspring, and you have something completely different. And that just starts
Hmm.
getting into how complicated these polymorphic type of gene combinations can become, because you really have a continuum of possible expression when you're crossing individuals, even if they look
Absolutely.
similar.
And personally, the way that I've come at that is to look for shades
Mm.
of color, because there really are, like, people will say, "Oh, I see yellow," and I'm like, "No, you don't." Like that looks like gold, right?
Yeah.
That looks like slightly orangey yellow. That, I
Mm.
mean, like, that looks like neon yellow. That's green.
Yeah.
That's like kind of on the green yellow spectrum.
Yep.
Like, there's all these. There's a whole lot of outcomes that you can
Yep.
see, and people often will just say the first thing is they, like, step back 20 feet away from the animal
Yep.
and say, "Oh, it's yellow," because they can see some from a
Yep.
distance.
Yep. But there's
Um,
muted tones of that, and that could be indicative of some of those genetic variations.
exactly, and that's, I think, what makes it really fun is when you get
Mm.
into the details and you're zooming in on every animal. You're trying to come up with a theory
Yep.
about what could happen, testing it,
Yep.
and then keep going, because it really is hard. Like,
Yeah.
I
Absolutely.
yeah. But that's awesome. I think that with the one of the things that I would hope to do long term is to get to a point where we can figure out what some of those underlying genotypic um, patterns are, right?
Yeah.
And we're very far away from that with
Mm.
pardalis, and that's why I personally is like some people are like, don't aren't you getting bored with the species? And I'm like, no, there's so much more to do. Like,
Yeah. Yeah.
um and what are your thoughts about some of the things that are there any species that have in captive breeding situation where we've kind of cracked the code, so to speak, on a more complex genotype?
I'm not as familiar with some of the other reptile species and their genetics. Ball pythons, of course, it seems to be a lot simpler
Yeah,
than chameleons. It seems to be individual genes, or three or four genes that you are crossing together to get fairly
certainly.
discrete results.
Yeah.
Um, when you start talking about some of the other gecko species, it seems like it starts getting a little bit more complicated, more going on. I don't know that there's any really highly polymorphic species that we've done a good job of really mapping it all out and figuring out all the combinations, the permutations, just because it's so complex. There's so many possible, combinations. I think ball pythons maybe is a good example of why that starts to become a challenging task, because when you start doing some of these crosses where you need three different genes to all have the recessive combination to be able to show this, super whatever duper,
Yeah.
whatever it is that breeder
Yeah.
is going for, the percentage chance of getting that is actually extremely low. And so that's why you have like these, double recessives, or these, triple recessives that are being bred back to, each other to try and increase those odds. And so for every gene that you add that you need, that specific combination, you make it more and more difficult to get that specific combination. And so
Hmm.
with something like Panther chameleons or something like that that has it seems like there's just a multitude of genes that are involved. The possible permutations are huge. You start getting like just um, astronomical odds to really get down to highly controlled genetics where you are producing really specific things intentionally. And so I
Yeah.
don't know that we have a great example of any of these highly polymorphic species that we've really gotten it down yet.
That sounds like a good challenge,
Yeah.
Professor Anderson.
Good luck.
So yeah, cool. Do you have any last thoughts for to leave people with?
No, I think, I give people a lot of credit that are working hard to work within these complicated you know um, gene spaces or color spaces to try and select for specific characteristics. I really like when I see people maintaining the localities from the wild rather than hybridizing across localities. But within those individual localities, there's a lot of potential there to emphasize specific aspects of those traits that are expressed to really see what the species is capable of. And I enjoy that. And I enjoy seeing progress that people are making, when they're working within that type of framework to try and see what they can do. It's fun to watch.
Yeah, it's a lab with that doesn't need funding.
Yeah,
So
yeah.
yeah, it's like that's how I think about it, at least. It's like we
There
can
you go.
explore so many cool things because we have revenue.
Yep. Takes a
So,
while though. Like different generate multiple generations. It takes some time.
oh, I know.
Yeah,
But thank you so much for joining us today.
no
This
problem.
is great. And I hope we can continue this conversation, and continue to pose questions as we keep going. So
Definitely, definitely.